My name is Zoe Watson, and I'm so excited to share my summer research project with you. This summer, I worked with Dr. Zardini Busato to conduct high-resolution mapping of brain lipid distributions in neurodegeneration. Lipidomics is a fascinating field in which lipids can be studied in many contexts. For this project, we were studying the clinical lipidomics of the brain. Lipidomics is super important in the study of neurodegenerative diseases because lipids can interact with many proteins which are involved in neurological functioning. The short-term aims of this project were to optimize a method of tissue lipid extraction and to map the spatial distributions of lipids across the midbrain, which is the region affected by Parkinson's disease. In this project, we wanted to try to map different classes of lipids across a space of tissue. Traditionally, MALDI-MS imaging has been used as the gold standard approach for tissue imaging and to identify metabolites, proteins, and lipids. In very simple terms, this process works by isolating a section of frozen tissue, spreading a matrix over it, and rendering images through an MS machine. Although MALDI has been a very useful tool, there are several areas in which it needs improvement, such as poor quantification accuracy. This is where our proposed method comes in. We use the novel technique of biopsy punches combined with liquid chromatography coupled with mass spectrometry, LCMS, to conduct sensitive analysis and more accurately identify the lipids across a space of tissue. Biopsy punches have thin stainless steel tubes connected to a plunger that are used for isolating tissue. We use these combined with a 0.5 by 0.5 millimeter grid to isolate as small as 0.01 milligrams of tissue. As lipids are fatty molecules, they can be isolated with an organic solvent, which was then analyzed by the sensitivity of LC-MS. In our optimized method, we first sliced the tissue using a mandolin to achieve 0.5 millimeter thick tissue slices. Then we use the stainless steel biopsy punches to extract small, uniform pieces of tissue into Eppendorf tubes. This is another angle showing how to use the biopsy punch. And here we can see the tissue that was isolated. To extract the lipids from the tissue, we used a modified Folk liquid-liquid extraction using 2:1 dichloromethane to methanol and a final wash with potassium chloride solution. Here we can see the phase separation. After lipid extraction, sensitive analysis by LC-MS was conducted to identify the classes of lipids across the space of tissue. This is a recording showing live results being output from the LC-MS machine, in which we can see specific peaks being detected. The following table shows the optimal conditions for lipid extraction as compared with initial conditions. Notable changes include using the wash step with KCl solution in replacement of water and increasing the sample to solvent ratio. The heat map shows that when using optimized conditions, all of the top 100 lipids were increased as compared with initial conditions. Additionally, this volcano plot shows that when using optimized conditions, 163 lipids were significantly increased. Imaging experiments prove that our proposed method of spatial mapping is indeed possible. This imaging experiment uses an overlaying technique of the original tissue image with the heat map of lipids to look at the spatial distributions across the midbrain. We can see patterns of alike lipids being in similar concentrations as we move down the column regions of the tissue. These results provide an excellent baseline of the distributions of midbrain lipids, which can be further analyzed and applied in regard to neurodegenerative diseases. From these results, we were able to determine that our short-term aims were achieved. Future work will improve and expand upon these methods into the neurodegenerative sphere. Next summer, we plan to apply this method to real brain samples with neurodegeneration, specifically Parkinson's disease, to analyze the spatial mapping of the lipids and their impact on neurodegenerative diseases. These are the references that I used for my research, and I wanted to thank Dr. Zardini Busato and the Lewis Lab for allowing me to use their space to conduct such novel research this summer. Thank you.